Microbial fuel cell for biotechnological education.
Okay, dear participants.
Your presentation on English or
Yes,
on Ukrainian?
on English. On English.
Okay, we'll wait.
Okay.
You can start, please.
Okay, so dear participants of the conference, I would like to present a report, Microbial Fuel Cell for Biotechnological Education. So actuality, modern biotechnology includes searching for alternative energy sources, one of which is the usage of microorganisms to obtain electricity. Even NASA microbiologists develop microbial fuel cells for prospective electrogenesis for future space flights. There are many forms of microbial fuel cells, which are abbreviated as MFCs, different in their scale. This theme is also included in syllabuses of biotechnological disciplines of many universities around the world. A lot of commercial educational models of microbial fuel cells were created too. However, these commercial devices require high level of investment. University and high school educators from developing countries seek alternative ways of creating microbial fuel cells from low-cost materials for educational purposes. Aim of our work was to construct a low-cost model of the microbial fuel cell created on the basis of a principal scheme. To do this, such tasks were fulfilled. First is we search for the principal scheme or model of microbial fuel cell. Second one is selection of the most optimal structural parts for it. Third was taking a sample of lake mud to make energy source. Fourth was assembling of microbial fuel cell. And the last one, fifth, was monitoring of electrogenesis done by microbial fuel cell. On this slide, you can see some examples of commercial microbial fuel cells. As you can see, their price is quite high for developing countries. So this was the main motto. We decided to create our low-cost model of it. Here you can see principal scheme of microbial fuel cell. As you can see, microbial fuel cell made of two chambers that are anode and cathode and proton exchange bridge which between them. Those chambers have electrodes immersed inside them. Electrodes are made of some conductive metal. Next, these electrodes are connected by wires to a resistor, and the resistor's voltage is then measured with help of multimeter or simply voltmeter. On this slide, you can see a model of microbial fuel cell we made. This device consists of cathode, which is aerobic, and anode, which is anaerobic chambers, which has 140 milliliters of working volume capacity, electrical wires, we used copper ones, and electron permeable bridge. Microbial fuel cell chambers made from parts of plastic pump. The content of a anode chamber was a source of bacterial electrogenesis. For these purposes, you can use river or lake mud. As for the cathode chamber, it was filled with boiled water, which was previously cooled to room temperature. As for electron permeable bridge, which is demonstrated by blue tube, it was prepared by using gel, which consisted of 10% of agar and 10% of sodium chloride in water. Microbial fuel cell was assembled by connecting this bridge between two plastic chambers, and after that the resistor was plugged too. In this slide, you can see results of our voltage measurements. As you can see, on 48 hours of exposure, we have the maximum increase of voltage level. It can be explained by the activity of mud bacteria in change condition. However, after 96 hours of exposure, we achieved drastic decrease of voltage. It can be possibly supposed that due to the fact that the resistor wasn't unplugged, it could freely spend energy generated by our microbial fuel cell. So, conclusions. As a result, educational model of microbial fuel cell is proposed. Positive results of its application were obtained, and maximum managed voltage, as was mentioned in previous slide, was 0.6 millivolts after 48 hours of exposition. This voltage should be enough to light up one low-power LED lamp. Several advantages of this model can be identified. First is low cost of consumables and perishables. Second is ability to make many copies of microbial fuel cells for several subgroups. And third is possibility to change configuration of microbial cells or contents of chambers to initiate more active electrogenesis. For example, to initiate ethanol or butyric acid fermentation, and to demonstrate it together with microbial electrogenesis. Thank you for your attention. If you have any questions, you can ask them now.
Thank you, Mihailo. Have you any questions, please? Yes, Dmitro, please.
I have one question. Can you tell me what electrodes did you use in microbial fuel cells?
Can you repeat your question once more, please?
Repeat, please, Dmitro.
What electrodes did you use in microbial fuel cells?
As electrodes were used bolts made of stainless steel.
Is it okay, Dmitro?
Yes.
Okay, thank you. Any more questions, please?